Hello, everybody. Thank you for showing up for the Wellick Lab meeting today. Just kidding. We do have two excellent speakers, both from the Wellick Lab, and I'm really excited to hear from them. I also did want to take a moment, though, to acknowledge both Muhang and Katharine for being good citizens of the genetics program. I know many of you are serving on committees as well, and I appreciate that as well, but I want to draw attention to Muhang has been serving as the chair of the graduate student committee for the last year, and Katharine's been serving on the diversity committee, and we really appreciate you putting that energy into the program. Thank you.
All right, great. Well, it's really lucky and exciting for me today that I get to introduce both of the students in my lab in the genetics department. So I think Muhang is going to speak first on her work. And Muhang graduated from Peking University, I'm not sure when, but she came to the university here and she is currently in 4th year. She's going to talk to you about done some really excellent work in the lab. She came into a project that was at that stage where it was almost done, but we could not get it wrapped up. And she also came in 2 months before COVID hit. So in addition to taking a really hard assignment, in some ways it's a gift, like here's 90% of the data. And then in some ways it's awful because it's like you clean up the last 10% and write it and get it out the door. And she did that and did a really beautiful job on that. And I think she's going to talk to you a little bit about that work. And then she's also been characterizing a new allele we made in the lab and focusing on starting some new projects, which I don't know if she'll get a chance to talk about. But Muhang has taken to heart something my lab always feels very proud of, and that is making very beautiful images. And she has taken that run. You're going to see that she generates just beautiful data. And I think that's an important part of telling a lot of stories. And so with no further ado, Muhang.
Thanks, Deneen and Nicole, for the introduction. Actually, the GSC doesn't have a chair, so I'm happy to know I just get promoted. Yeah. And today my topic is to understand the function of HOX5 genes in the lung. And I asked myself, you might also ask me, why am I doing lung research? So we know that lung is important. We use it to breathe. And throughout history, there are so many different lung diseases that are fascinating and intrigue all the scientists, patients, and doctors, such as asthma, COPD, lung cancer, and something you might not heard of, COVID-19. In the CDC database from 2019 to 2020, top 10 leading causes of death in the US, 3 of them are related with lung diseases. So I think it's fair for me to say that understanding the lung development, homeostasis, and disease process is very important. And I'm from Wellick Lab. In Wellick Lab, we study HOX genes. What are HOX genes? They are transcription factors, the encode transcription factors that direct the development of body segment in animals. You can think of them as these little instructors. When an animal body is building, and they will say, you put a head there, you put a tail there, and the limbs here. And if HOX gene messed up, bad consequences, you can guess. In mammals, mouse and humans, we have 39 HOX genes. They located in 4 different chromosomes and forming 4 different clusters, A, B, C, and D. And mostly focusing on HOX5 genes, the 3 of them, A5, B5, and C5. They are known to be crucial for the lung development. In the past, Wellick Lab find out that in mouse embryo, so E18.5 means it's the embryonic day 18.5. If we lost all of the HOX5 genes, A, B, and C, a very beautiful embryo lung turns into this abnormal, severely hypoplastic lung, and this mouse will not be alive. At postnatal stages, loss of HOX5 function leads to a BPD-like phenotype. So BPD is bronchopulmonary dysplasia. It's a condition often found in premature human babies, and they have larger, fewer alveoli, and makes them harder to breathe. And this is also seen in our HOX postnatal, mutants. You can see it in the wild type, the alveolar are, there are more little surface for the mouth to breathe, but the wall is thinner and there are less air surface. So HOX genes is actually the most studied genes. No, the oldest to be studied genes in animal development. But it's not very well studied in adult. And scientists used to believe that HOX genes do their job when the body is building, when the body is built, they're done for their job. But that's actually not true, because recently a lot of work shows that HOX genes are involved in adult homeostasis. And that's also my first project, which is trying to see if HOX5 genes also function in adult lungs. And that's the one that Janine mentioned that I'm helping to wrap up. And this work is published last year. I'm just going to show you some highlight of my results. When we knock out HOX5 genes in adult mouse lungs, we also see this enlarged airway in the mutant. Sorry. In the mutant, just as we see in the postnatal animals. Also, we see a breakdown of this here showing green elastin extracellular matrix in the lung. This is quite surprising because the elastin network in both human and mouse lungs are believed to be extremely stable. Their half turnover time is 74 years. So we're surprised to see that there's changing that caused by HOX5 deletion. When we isolated fibroblasts from mouse lung and put them in vivo conditions, they exhibit reduced adhesion. Well, this all looks very nice and is published, but we still don't know what HOX5 is really like causing all of these phenotypes from the embryonic stages to adult stages. So the deep mechanism we still don't understand. And that's my second project, which I'm trying to see which type of cells in the lung are contributing to all of these phenotypes, the enlarged airway, loose adhesion, and exactly what cells in the lung are really expressing HOX5. So we do know that in embryonic mouse lung, HOX5 only expressed in the mesenchyme and not in the epithelium. This is an in situ hybridization assay. You can see that, you probably can't see that in the projector, but the purple thing is the expression of HOX5. It's mostly in this mushy, lot of cells. They are called mesenchyme, but not in this thin layer and forms a ring called epithelium. And that's basically all we know. And apparently this is not detailed enough for me to answer my questions about HOX5 function and to really monitor cell like HOX5 activity in the cellular level. And that's why our lab designed this new reporter line, HOX-A5-EGFP, that allow me to answer these questions. In the wild type A5 allele, there are 2 exons. And in the GFP allele, we swapped exon 1 to an EGFP cassette. So wherever A5, HOX-A5 expressing, we should be able to see the green signals. And I got this line, and I need to validate it to see if it's working, if the green signal is there, or if the signal I see is where it should be. And first, I'm looking at it in whole mount embryos. as early as E9.5 to E16.5. And I'm happy to see that they are very consistent, robust, and intense HOX-A5 signal at where it should be in the hindbrain, in the spinal cord, in the scapula, and those are recapitulated what has been reported in the previous published literatures. So into more details, I also look at HOX-A5 expression in sections. So this is a embryo frontal section. It's like section this way. And I see HOX-A5 expression in all of the respiratory tract mesenchyme, trachea, bronchi, and lung, and not in the epithelium. It's also seen in the diaphragm. And looking at the digestive system, I see expression in the intestine, mesenchyme, and the stomach. More specifically, here is a cross section of the digestive system. Cross section is section this way. I can see the expression in the duodenum, in the midgut mesenchyme, in the stomach mesenchyme is very bright. And if you like pancreas, you know that PDX1 is a pancreas epithelium marker. It's like a flower, like two flowers. And I also use this antibody to help me identify pancreas, and I see the expression is also in the pancreas mesenchyme, but it's not in the hindgut. So all of this is corresponding with what we believe, like what we already know. I also look at other systems like skeletal system and central nervous system. And just briefly tell you, I see the expression in the spinal cord, in the vertebra, and also in the numbers that are in the cervical and thoracic region, which is also HOX-A5 should be in a lot of bones near the sternum area. So all of this proof that this most likely this line is correct, is accurate, is working. So I can really start to use it to answer my question is to define lung cell types that express HOX-A5. But before that, I need to know what cells types are in the lung. And that's a very big question. So just to simplify it, I'm grouping cells into 3 major groups: epithelial cells, mesenchymal cells, and endothelial cells. Here is the alveolus, and I'm focusing on 2 epithelial cells. The epithelial type 1 cell, or ACC1, is this thin, large, abundant cell that allow air exchanging. And type 2 epithelial cells is this more rounded and less abundant one. In the mesenchyme, we are focusing on fibroblasts because those are the cells that we see phenotype in our mutants. And I'm mostly staining for 2 different types of fibroblasts: myofibroblasts and lipofibroblasts. Oh, sorry. And also endothelial cells in the blood vessels. So I'm using different antibodies of specific cell type markers and to stain those antibodies in my lung sections and to see if I can see overlapping HOX-A5 EGFP expression. I'm doing this first in postnatal animals, and I want you to appreciate that in the whole month, even I can see that my postnatal GFP line is also still very bright. So there's a lot of HOX-A5 expressing in there and not in the wild type. Looking to sections, I see the same thing. And first, I'm looking at HOX-A5 EGFP co-expressing with, staining with T1 alpha positive type 1 epithelium cells. I know this is hard to see. It's difficult for me too. Lung, due to its histology, is kind of messy, and it's hard to figure out like cell borders. So that's why I'm using DAPI as a nuclei dye to try to help me see each individual cell And this is a zoom in of a P1 alpha type 1 epithelial cells and a HOX7-VEGF cell. And mostly I don't see them overlap. A similar thing I observed in the type 2 epithelial cells. So I don't see them overlapping. And when we jump to mesenchymal cells, first I'm looking at alpha SMA positive myofibroblasts. And here I'm staining it in red. I'm also staining T1 alpha, the epithelial cells, in white, just to show you a border and the shape of the lung. And anything you see orangey, yellowy are overlapping of myofibroblasts and HOX-A5-VEGFP. Similar things are seen in the ADRP positive lipofibroblasts. We see a lot of overlapping expression. Well, and this is something I just found out today. And I was surprised by this result, which is I found HOX-A5-VEGFP co-expressing with PCAM positive endothelial cells. Like previously, we think they are not in the endothelial cells, but when I do look at this blood vessel, I can clearly see there are 3 cells. This is a nuclei, and PCAM is a cell surface marker, so it will not show in the nuclei or the cytoplasm, but it's definitely the same cell as the HOX-A5-VEGFP cell. And this is preliminary data, and we're gonna do more, maybe other stainings and look closely at more sections to try to confirm this. And so far, I did this staining, and I did see overlapping in mesenchymal and endothelial cells with HOX-A5-VEGFP in postnatal lungs, and I'm gonna do perform this in adult lung in the future. And so far in the adult lung part, I see GFP signal in our GFP animal, but not in the wild type animal. And be able to see that give me promise that I can repeat all of that in my adult animals. I know that it would not be super easy because adult lung is not very easy to work with. There are a lot of autofluorescence, and the cell types are even harder to find, but I will try to make that work. And I think this can give us more information about what cells are expressing HOX-A5 and try to help us understand the exact function of HOX5 in the lung. And also, this is only a part of my project. And due to time, I'm just going to talk about some R&R today. So in Willick Lab, we have a lot of transgenic mouse lines. And if you have a lot of mouse lines, you need to do a lot of genotyping every day, daily. And it's trivial, but it's also super important because the worst thing can happen to a mouse lab is you mess up with your mouse line. And that's why every time we do a genotyping assay, we will have a positive and negative tail control and replicates of our mouse tail samples. We also refresh our mouse line regularly to backcross them to the appropriate strain to maintain their health and make sure they're fertile. I'm also including both male and female in my studies to avoid any sex bias. And I'm always using litter mate controls if possible in my studies. For example, in this HOX-A5 GFP project, I can see there are no GFP signal in my negative litter mates, wild type litter mates. I'm pleased with that. And thanks for Willick Lab. So yeah, that's the name. And yeah, we have a lot of people in the Willick Lab, and we study 2 HOX genes, HOX5 and HOX11, and I'm the only one who's studying HOX5. And Kat, who studied HOX11, will show you her work. Oh, question. can't see chat from here. Dude, is there any chat in the Zoom? You mean after I did that? Well, well, that's a good question. I want to go find other resources. Like I'm only doing staining here, but I know there are single cell sequencing of mouse lung, and I need to like confirm if they see the same HOX-A5 expression in a single cell sequencing. And then I want to publish the result, but also I am doing this RNA-seq in my postnatal and adult mutants and wild type to help us to find some downstream target gene candidates that get up or down regulated after HOX5 deletion. And just to study all of these different perspectives can help us to map those informations together and hopefully understand more. Yeah, that's a good question. I'm also thinking about what I'm going to do after. So yeah. Yeah. I don't think each of them are easier or harder than the other one. Staining takes time and is pretty, and I think flow is doable if we design the experiment very well and choose specific cell surface markers. But also a lot of cell types I'm showing here, they don't really have a marker that can really identify them in vivo, but in vitro, we can at least see the shape and location of the cell. So we know that it's an epithelial cells and it's at where it should be. But in the flow, you can only trust your antibodies. And I never done it before. I know it's kind of tricky, but I think it's interesting to do just to confirm if the flow and staining have similar results. Oh yeah, I didn't talk about it here just to make it simpler, but so actually the deletion is a time and location specific deletion. So we flux the both HOX-A5 and use TBX4 is a long mesenchymal specific enhancer to drive a crate. So when we give the mouse doxylin, the crate will induce a deletion of HOX-A5. And we are dox-treating them at age week 8 to week 10. And I collect my adult animals at week 10. So it's definitely adult. And the deletion is also happening in adult. Yeah. Right. And lung is believed to be developed at postnatal day 60 or week 6. Yeah. Yes, it's homeostasis, not development. Nicole.
This as you will, being a bacteriologist, I don't spend much time looking at histology of lungs, but it looked like in some of your images that not all the cells of the mesenchyme were shown in GFP. Cells that are and that aren't. It looked like some of the cells were just white, not green.
Yeah.
The epithelium, but it also looked like in the mixture there was a sort of heterogeneity.
It could be like there are so many cells in the lung, and it could be like macrophages, and
I think there's some other
yeah,
a mixture of cell types there, some of which
yes,
are
yes,
and some of which are not.
right, yeah. Also fibroblasts, there are different type of fibroblasts, and nobody really know how many types are there. So yeah, the difficulty of this research is I want to define the cell types, but the cell types are not defined. Like what cells are in the lung is still being studied.
So you're doing single cell genomics or bulk graph?
Bulk
So-
mesenchyme
Yeah. So
fibroblasts.
given then that you have a heterogeneous population, how are you going to approach interpreting that data? You know, if you have some cells that are box expressing and some aren't.
Yeah, we in this sequencing, we isolated fibroblasts from lungs and culture them to passage 3 and then bring the cells to RNA-seq. And when I culture my HOX-A5 GFP adult lungs, so not this animal, GFP, I can see that most of them are green in the culture. So like 95%. Yeah. But I think it's very complicated, the question you are, like what we are doing here. Not yet. Well, I can see that even in alpha SMA, myofibroblasts, not all of them are HOX-A5 GFP positive. And there are some other markers I can look at: PDGFR alpha, TCL, F4. Yeah, we could look at them.
I have one more question. Which I know it's hard to answer without getting this data first, but I understand. So in the adult lung, you're looking at homeostasis, not development. But what kind of role could you imagine? You must be anticipating what would homeostasis mean in this case? What sort of homeostatic role could HOX5 be?
So we have clues and inklings that HOX5 and other HOX genes are functioning in cell and cell matrix adhesion, like cell behavior, cell adhesion. So we think that's probably has something to do with it. Also, we see the elastin phenotype, and that's basically what I can tell you.
So your expectation when you get back your RNA-seq is you do enrichment analysis and
right.
Cell adhesion is going to pop up.
Yeah, we're going to look at cell adhesion. If we do a geo analysis, and we're going to focus on adhesion. Yeah. Hi.
Hi, great talk, by the way. Are there good alveolar epithelial Cree drivers available? We're looking at mesenchyme.
Yes. Why?
The lining of the is epithelial, right? So
yes.
Do I not understand this?
Yes. I think there are. So you think I can use that?
I'm just asking because there are multiple tissue layers here.
Yes. So HOX-A5, we're focusing on mesenchyme.
Right. I'm just asking for someone else who comes into the lab, or are there good Cree drivers?
Oh, not in our lab. No. Yeah. Sorry about that. Okay.
Thanks. And next you'll hear from third year graduate student Katharine Hubert. So Katharine joined my lab, and I said, I have this really cool idea based on data we've been generating for this project, but you're gonna have to learn to fracture animals because the collaborator mission is to do that. And we don't really have anybody here to do that. And I think single cell seq, which I've avoided like plague, is a really perfect way to answer the question. And we've never done that either. And I think it would be better for your career if you learn the bioinformatics yourself, although we have a collaborator. I think that would be a really great thing for you to be able to do this project forward. So in the last 2 years, Katharine has accomplished an incredible amount on all of those fronts. And I'm excited to see what the next months and years for her project show, but I think she's making great headway on what I think is an interesting story. And so Katharine, give us today's update.
Can you guys hear me okay if I talk like this? Okay, great. So unsurprisingly, we're going to continue to talk about Hox genes. Part of the reason that Muhang and I both decided to present today was not only because we thought it would be fun, but to really highlight what these genes are doing in different contexts and how we're both trying to address the question of what Hox genes are doing in adult animals, but by different systems and mechanisms. And so Muhang briefly mentioned that in mammals, there are 39 Hox genes that are divided into 4 chromosomal clusters, A, B, C, and D. And these genes are further subdivided into 13 paralogous groups that arose due to genome duplication events. And so these transcription factors that are really important during development have interested biologists for 3 really unique reasons. The first is that they have a clustered genomic arrangement. So within a given chromosomal cluster, these Hox genes are all together, and there are no non-Hox genes in between them. So one interesting property. The second is that they exhibit what's called collinearity, and that means the order at which they're expressed during development mirrors their position on the chromosome. So for example, Hox1 genes are expressed first, and they're expressed more anteriorly in the body. As development proceeds, the Hox complex is progressively unwound as the body axis develops, such that Hox13 genes are expressed last and most posterior. And finally, Hox genes are spatially restricted. After they're deployed during development, they're expressed in a specific region, and that region is maintained through adulthood. So naturally, developmental biologists were very interested in this unique set of properties that Hox genes have. And scientists do what scientists do best when they don't understand what genes do. They knock them out, study the consequences. So here we are looking at the mouse spine. And so in the mouse spine, we see that there are ribs in the thoracic region, as we would expect, but there are no ribs in the lumbar and sacral regions. When we generate a Hox loss of function mutation, you can see that these ribs now extend into the lumbar and sacral regions. And this is a property of Hox genes that result from these mutations called homeotic transformations. And a homeotic transformation is when the mutation causes an anteriorization of the skeletal elements, such that they adopt the identity of the element anterior to them. And so in this case, the lumbar region now has an identity similar to the thoracic region, where ribs now protrude from that area. Similarly, a subset of Hox genes, specifically 9 through 13, posterior group, are expressed during limb development and help to pattern the proximal distal limb axis. And so specifically, Hox 9 and 10 is expressed in the stylopod. Hox 11 is expressed in the zygapod, which is the radius and ulna or tibia and fibula. And Hox 13 genes are expressed in the autopod. And so just like the anterior posterior axis, if we were to create mutations or knockouts of these genes, we also see defects in skeletal patterning. So here I'm focusing on the Hox 11 paralogous group of genes. You'll notice down here that there are 3 members, A, C, and D. However, in the forelimb, only Hox A and D are expressed. And so if we generate knockouts of these animals, here indicated by lowercase letters, you see that the zygapod region is mispatterned. And so collectively, by generating these mutations, we've shown that Hox genes affect skeletal patterning and identity. So it would be natural to think that these genes play a role in development of the bone and the skeletal system. But early work in the lab found that HOX11 is not expressed in bone or cartilage cells, and bone and cartilage is what makes up the developing skeleton. And so to study this, we generated a HOX-A11 eGFP reporter that allowed us to visualize HOX11 expression in cells by green fluorescence. We also found in this study that HOX11 expression was on much longer than we anticipated, meaning the embryo was developed and we were still seeing HOX expression. So we decided to see how long that expression lasts, and it's maintained through adulthood in region-specific areas of the forelimb, specifically the bone marrow, the periosteum, and the endosteum. And so the periosteum and endosteum are connective tissue layers that envelop those regions of the bone. So now we see that HOX11 expressing cells are maintained through adulthood, but what are they doing in adults? In order to answer that, we generated a HOX-A11 Cre-ERT2 allele, which allowed us to do lineage labeling, such that cells expressing HOX-A11 Cre are red, and every time they divide, those cells are red, and they stay red. So we can track where those cells go in the context of bone homeostasis throughout life. And results of this study showed that our HOX11 expressing cells gave rise to bone, cartilage, and fat. So they were capable of trilineage differentiation in vivo and solidified the notion that HOX11 expressing cells are indeed a skeletal stem cell population. But what are they doing in the adult skeleton? What is happening here? And so if you think back, and I think Xiaoya asked a good question about this, how do you study these genes in adult mice if when you delete the genes, you have developmental defects? So like Muhang, we generated a conditional deletion allele here in HOXD11 though. And so here LOXP sites are around exon 2 of HOXD11, such that when we give the nice tamoxifen in combination with rosacree, we can control when we're deleting these genes. And so for the context of my experiment, we give the mice tamoxifen chow from 8 to 10 weeks of age to enact deletion, and then continue our studies with the adult mice. And so this study really showed an interesting finding. Just to orient everyone, CB is cortical bone, endo is short for endosteum. And so what we see in the control mutants is that both of these animals express RUNX2. And RUNX2 is a transcription factor that marks the commitment to the osteoblast lineage. However, the mutant cells don't express osteopontin or osteocalcin, suggesting that they turn on RUNX2 and start differentiation to osteoblasts, but that they're not able to complete this differentiation. So it's perturbed in our adult homeostatic model. And all of this leads to my question, which is how HOX11 genes are regulating repair and differentiation in the adult skeleton. And in order to address this question, I have to use the in vivo fracture model that Denise mentioned, because it's the only point in adult animals where we can simultaneously observe differentiation into both bone and cartilage cells. And that's because bone heals through two main processes: endochondral ossification, which is where the stem cells differentiate to a cartilage intermediate before bone to remodel the matrix, and intramembranous ossification, which is a direct differentiation of stem cells to bone. So using this model, I am hoping to address two questions. The first is, what impact does loss of HOX11 function have on the ability of animals to repair fracture? And then secondly, I'm doing single cell sequencing to look at the transcriptomic changes that are occurring as these cells differentiate to bone and cartilage. But before we get into my actual work, I think it's important to understand what is normally supposed to happen in the context of fracture repair and healing as sort of a basis for what to expect. So immediately after fracture, a hematoma or blood clot forms to stabilize the fracture site. There's a lot of inflammatory markers that are gradually resolved by 5 to 10 days post-fracture. And it's at this time point that we're most interested in. If we jump over here, I think this died. Oh, it's still there. Oh, well. So at 10 days post-fracture, this is where we have the simultaneous differentiation to bone and cartilage. And so chondrocytes or cartilage cells are primarily located in this central region immediately adjacent to the ends of the broken bone. And during endochondral ossification, their role is to provide this preliminary scaffolding to work during the repair process that's gradually replaced by bone as time proceeds. Away from the central callus region, we have differentiation of stem cells to bone cells directly. And so in those regions, when I'm looking in my fracture slides with the immunofluorescence analyses, it helps me to know where to expect these populations to look for differences in healing. And so at 3 weeks post-fracture, this cartilaginous callus here should gradually be remodeled and turned to bone, such that by 12 weeks post-fracture, we expect healing in wild-type animals where the bone is completely remodeled. So before I did anything else, I needed to validate that my mutants did indeed have a phenotype. And so I performed the ulnar fracture surgery. And in this surgery, the ulna is fractured, a nice transverse fracture through the ulna, and the radius is left intact to stabilize the fracture site. Then these mice are taken out to 10 days, 3 weeks, 6 weeks, or 12 weeks post-fracture to assess healing. And so in our control animal, you can see it's a very good fracture. But by 6 weeks post-fracture, that line's no longer visible, and the control is remodeled fully at 12 weeks post-fracture. If we jump down to the mutant, you can see another great fracture through the ulna. However, at 6 weeks post-fracture, the bone looks a little circular and swollen, and that's not resolved by 12 weeks post-fracture, indicating that a callus is likely present and that mutants do not heal by 12 weeks like they should. So having established that the mutants do indeed have a phenotype, I then went on to look at or start examining what could be going wrong at earlier stages of differentiation, specifically starting at the 10 days post-fracture time point. And unfortunately, this projector does not do my images justice, but bear with me. Right here, this circular area is the fracture callus, and the white dashed lines outline broken bone. And so both control and mutant animals express RUNX2, which is that marker for committing to the pre-osteoblast lineage. They also express ostrix, which is a transcription factor marking an immature bone cell state. However, mutants fail to express osteocalcin, a marker for mature osteoblasts, suggesting that these cells initiate differentiation into bone, but that they're not able to fully mature. And so likewise, I've started to look at differentiation into the chondrogenic lineage. And so far, I can tell you that both mutants and controls are able to initiate this differentiation. And this is visualized with SOX9, which is a transcription factor marking commitment to the chondrogenic state. So we see that both of them initiate differentiation into the chondrogenic lineage. And while I'm still collecting data on this, preliminary analysis indicates that the mutants retain a cartilaginous callus at 6 weeks post-fracture, way later than they're supposed to. And so this is histology with Sappho fast green staining. So the bone is this teal color and blue. The red here is the cartilage population, and then the nuclei are marked with hematoxylin. And so we can clearly see in this mutant animal the presence of the red callus still in the middle. Furthermore, another interesting observation is that you can clearly see both ends of the bone where it was initially broken. So it's not being remodeled appropriately like in the control. You don't have evidence of that clean break at this time point. So while this data is preliminary, it is certainly interesting, and I'm working to collect more on this. So switching gears a little bit, to supplement this work that I'm doing with the in vivo model, I'm also doing single cell transcriptomic sequencing, which will help me to further that idea along as well. And so this model is quite complicated, so I'm just going to take a minute to walk you through what's happening. At homeostasis, as I mentioned before, our HOX11 stem cells are in the bone marrow and in the periosteum, the outer connective tissue layer. And this model stem cells are marked by GFP, so any cell that's expressing HOX11 GFP is green. We also are using the HOX11 Cree ERT2 in this model in combination with Rosa TD tomato. So these cells also have the ability to be lineage labeled, which means that when we give the mice tamoxifen, stem cells that have successfully undergone this recombination to remove the stop codon will be red and green. As these cells differentiate from stem cells, they'll turn off HOX11 GFP, no longer express that transcription factor, but will stay red. And so we'll be able to see what these cells are becoming. And so at 10 days post-fracture, this is a representative model of the callus. You can hopefully see that we have a population of red-green cells representing stem cells and just red cells representing cells at various points at differentiation as they become bone and cartilage. And so using FACS, we collect cells that are red-green and red to submit for single cell RNA sequencing. And this model is extremely powerful because it won't only allow us to look at the transcriptome of the stem cells, but also all of the cells at various points in the course of differentiation into both bone and cartilage. And so this really gives us the best shot at identifying downstream targets of Hox genes. So, wow, this looks bad. For the sake of time, I'm not going to explain all the analyses that it took me to get here, but I will just briefly orient everyone. There are control and mutant samples in this population. We're looking at a UMAP, which groups cells based on clusters of shared gene expression. And so this is looking at the population of all cells when we input them into this analysis, but we're only interested in certain cells. We know that Hox genes don't interact with immune cells or aren't from the HSC lineage. So those are cells we'd want to exclude for future analysis. And so basically all of these black boxes indicate cells that I'm excluding so that we can just focus on stem cells, bone, and cartilage. And this is supposed to show you that there's a relatively equal distribution of control and mutant samples in the populations that I'm excluding, which is important. We don't expect there to be a difference anyway. So now we're looking at the resulting UMAP when I only retain bone, cartilage, and stem cells. And right off the bat, when you compare this map to the UMAP showing the controls and mutants, you can see that there are obvious differences in cluster compositions between these samples. So to highlight a few, 63% of the control cells fall into this grouping. And this grouping, while I'm still working to discretely identify each cell type, I know to be associated with differentiation into bone and cartilage. They have those markers that I was showing you earlier that we would expect to see in this area. So 63% of the control cells are differentiating as we would expect to bone and cartilage. However, only 9% of the mutant cells fall into this population, which means that 69% of the mutant cells are in this large grouping here. And so while I can't, I haven't identified these all yet, but obvious next steps include figuring out what on earth this cluster is. It seems to be the key to what's going wrong as the Hox cells differentiate. But also one other thing I really want to do with this analysis is lineage mapping with Monocle. And so Monocle is a algorithm software that allows you to do pseudotime analysis. And so it sort of plots the course of differentiation of these cells as they go to bone and cartilage based on gene expression values that are changing with respect to time. And so we expect that there are differences in the lineages of controls and mutant cells, and Monocle will help us visualize that. So just to summarize what I've shown you so far, ulnar fracture repair is abnormal in HOX11 mutant animals. At 10 days post-fracture, these animals exhibit defects in osteoblast differentiation. They initiate it, but the cells aren't able to fully mature. And my preliminary single cell sequencing results suggest that these mutant cells have an alternate fate. And before I take questions, I just wanted to mention rigor and reproducibility, because that's really important to all science. For animals, I make sure to use age and sex matched animals. Sex of the animal can influence the size of the bone, which is important. I also try to use litter mates when the odds are in my favor. Sometimes one of these mutant animals is a 1 in 16 chance of generating, so that's not always the case. But we do annual outcrossing to maintain the genetic background, especially after these animals have been homozygous for a while. Experimentally, I pay very close attention to fracture type and pattern. Early fractures that I did while I was still developing the technique showed that if the fracture is diagonal or oblique, it doesn't heal at the same rate as the transverse fractures that I showed you did. So oblique fractures are not good for long-term healing studies. There are obvious differences there. We also validate the allele deletion after treatment with tamoxifen by genotyping. So I'll take a snip of the ear and do PCR to check to see if the recombination event happened as we expected. It's also important that I compare bone sections at similar depth. So the callus is a 3D structure, and if I take a section off the top, that composition of cells is not uniformly distributed throughout. So making sure that I'm looking at the middle sections for the most important parts of my analyses, because the middle sections have good populations of bone and cartilage. And finally, I use a lot of antibodies, so I keep a good record of the catalog number for future use for both myself and any future lab mates. And so with that, I want to thank my lab. A special shout out to Corey, who assists me with anesthesia for all of these surgeries, makes the process go a lot faster. My thesis committee, funding, and my former lab. And I'm happy to take any questions. Yes. Hi, Sarah.
So earlier you talked to earlier in the talk you stated that in the working more are there any humans associated with the HOX11 mutation?
Yes. HOX11 during development affects the kidney. So actually in mice, when we create these mutations, they're not viable after birth because of the kidney defects. Other HOX mutations in humans, I believe there are 14 syndromes. Most of them are associated with the posterior HOX genes, and they'll present with obvious abnormalities like polydactyly or syndactyly or brachydactyly, or craniofacial defects. And we suspect that there are more human conditions that are a result of HOX mutations, but for example, if you have a mutation in HOX5 and it's a defect in the lung, that's not something that's obviously visible, whereas a skeletal abnormality is, so they may be missed. They're also upregulated in a lot of cancers. Mm-hmm. Yes. Oh boy. 10 days post-fracture. At that time point, we have good differentiation into both bone and cartilage that's relatively balanced and not skewed one way or the other. Yeah, no, that's a really good question. So I think those are both good hypotheses. One could be that it is stalled or stuck at a certain point. However, preliminary analyses indicate that this population and this population are both expressing genes associated with chondrogenesis. So they're at least starting that expression. However, this mutant population also has high expression of a lot of the markers associated with inflammation. It's almost all preliminary, of course, taking on an osteoarthritic type phenotype. Yeah, the cartilage. Yeah. One possibility, or it's doing something completely different with cartilage. And I have a crazy hypothesis for that, but I'm not going to say it. But I think, seeing that this bone fracture is still visible here could explain the widening. That is something we've noted. Something else I've been looking at are defects in the periosteal matrix, which are apparent in mutant animals at homeostasis. So the matrix, the periosteal matrix that surrounds the bone is where the stem cells are embedded. And in mutants, that is disrupted. So that could be, affecting how it's the ability of cells to differentiate. Oh, yeah. Yes. So also, another reason to choose that time point is that we have a lot of inflammation at earlier stages of fracture healing, and we don't care about immune cells. We don't want them in there. We got some, but then we excluded them. So one. And two, we need to have a cartilage population laid down, connecting the bones in order to see matrix remodeling happen. And so if we were to choose a slightly earlier time point, the cartilage matrix may not have had an opportunity to have been laid down to begin to see that active process of remodeling at the 10-day time point. I don't know if that. Oh yeah, that too. That's important. They're still proliferating at 10 days. Yeah, which is really important because we want to collect stem cells too, not just the differentiated cells.
I would think because if they were stalled, they would end up in the group.
Yes. Maybe here.
Because of all of the stages.
Great. Yes, sorry, I did not articulate that.
But
I love parallels. I think early on you said two of the parallels are expressed in this area. So why did you choose this one to pursue, and why you did the same experiments to knock out of the other one? Would you expect to see the same effects or something different?
Yeah, so during development, only HOX A and D are expressed in the forelimb, whereas HOX AC and, or sorry, HOX A, yeah, AC and D11 are expressed in the hindlimb region. And so we picked this because you only have to knock out 4 alleles instead of 6, like if you added the C per allogous group. So it's just faster and easier genetics.
You're actually knocking out both.
Yeah, all of HOX A and all of HOX D11 are knocked out. Yeah. And if we were to use the hindlimb, we'd also have to knock out HOX C11, which is just more work.
So you think they're entirely redundant? Does the single or knock out just one or the other? Do you get these effects?
I haven't, so during development, if you just knock out one, the bone forms as expected. So in that sense, yes, they're redundant. I haven't aged those mice to adults myself personally, but also studies, there were HOX swap studies. So if you swapped exon 1 of HOX A11, for example, with exon 1 of HOX D11, it was able to function normally in that sense.
In development, right?
Yeah.
Has anybody ever looked into the development? You could have the parallels further.
Oh, she did? Oh.
Divergent function.
Oh yeah. Yeah. So one of the lab mates for the study that validated the HOX D11 allele looked at genetic controls with knocked out HOX A function as a comparison to the conditional deletion, and they were fine. They were normal. Yes. Sorry, it's hard to hear you, but I think you asked if I see an accumulation of cells in the control, but I don't. Oh. No, there's no evidence of. I'm not sure I fully understand the question, but at in vivo, there's no abnormal differences in stem cell proliferation. Those both occur normally. There's no certain accumulations that indicate fibrosis at any time point in controls during the context of fracture healing. I haven't looked at mutants at, for example, 6 weeks post-fracture to see if those cells are accumulating in a region and representing a fibrotic phenotype. I'm not sure if that addresses your question, though. Oh, I see what you're saying. From what I've looked at, no.
Was one of your early slides, I think, was it an X-ray maybe?
Mhm.
Very early where you showed what the break looks like. The pseudopod just looks stumpier there. Is that typical in the bottom row, or is that just an accident in particular?
No, so the mutants have, you see how the radius is very bent. That's one of the hallmarks of the mutation in adult mice, and it's also shorter. So it is stumpier. We've taken measurements as I fractured. The controls average about 14 millimeters in length, whereas the mutants, if I'm just going from here to here, not taking account that bend, are shorter, closer to 10. So yes, and also there's no scale bar here, which I should add in the future for better rigor to illustrate that.
Is that a difference in epiphyseal growth or prior to fracture, or like why are they stumpier? Is that understood?
I don't know. I honestly don't.
Because there's a lot of, like, I know, I'm sure Deneen was probably on a lot of these papers with all these Alcian blue stained skeletons look really beautiful, but I don't know anything about the histology myself. I just wondered what is much known about.
Yeah, I'm not
That is
sure.
kind of a follow-up. I think that was Francisco back there. Kind of a similar question.
Yeah, I'm not sure why they become stumpy, but it is a consistent phenotype in our mutants. Do you know why? Oh,
Well, this has been deleted for 12 months already, so fracture rate is also not continuing to grow. So Jane when you stabilize fracture, the bones will start to bend.
So there's a mechanical
Everything about
component
that
to
is
it. Okay.
exacerbated.
An optical illusion, or is that impacting the other bones when you grow?
yeah, no, I definitely broke this one.
Yeah, I can see that, but yeah.
It's possible that, hey, I don't do this under a microscope. I'm basing this all off of feeling, and to just be able to fracture one is hard in and of itself. So it's possible that it nicked the radius but didn't break it. Or that, like I said, there are defects in the periosteal matrix, so that during this remodeling process, it's not doing that appropriately in containing cells, but that's preliminary. I haven't specifically looked at that to see if there's a connection with that gap. Yeah.
Like when you got this, it looks kayak style, like if you back and maybe that's maybe that's also upgrade healing. And so I know you like food and new power.
No, but maybe is my answer. We know Hog cells are derived from the mesenchymal lineage, not the HSC lineage. So first, we really don't care what the immune cells are doing. But yes, and that is likely autofluorescent contamination from sorting. However, I will say that differences, if there are any in immune cell populations, could be informative as to what's happening upstream, not directly caused by Hox genes, but could help explain what is wrong, if that makes sense. Like if we see a certain set of well, we did Danielle published when she first fact sorted. No, I think that's something different. But when we first established that these cells are stem cells in the bone marrow, Danielle fact sorted and excluded HSC lineage cells. Hox was not expressed in that lineage in the bone marrow based on cell markers during fact sorting. Does that answer that? Another question.